Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. We have been working on a RNA-seq data set and we have gone through several steps, right. So, we have started with the count data and then we have done some preliminary analysis through distance based clustering and principal component analysis. We have seen how the samples are similar or whether there is an outlier, right. So, we have found one outlier in our sample and then we did the bias correction using the package EDSEC. We corrected for GC content, but of course, you can try different ways for this bias correction or within sample normalization. So, now we have the bias corrected data and we can move on to the differential expression analysis, the actual differential expression analysis, right. So, in this class we will be talking about this expression analysis using this tool DSEC2. Okay. So, this is the agenda for this class. We will talk about the first part, we will cover the first part of this differential gene expression analysis. There will be a second part where we will cover this differential expression analysis when you have the spike in samples. Okay. So, the normalization we can do differently and that will cover in the next class. Okay. So, again coming back to the steps, we have completed up to this bias correction part and we can start with the differential gene expression analysis, and for that we will use this tool DSEC2 that is I have already introduced and I have also shown you, right, how we can install this package. Okay. So, we will now use this package to actually run this analysis and then of course, we can generate some interesting results which we can interpret and visualize. Okay. So, let us now move on to the R console where we can actually load the data, the sample information and we can proceed with the differential gene expression analysis. So, we will go back to this folder where we have all these files, right, and as you remember we will now we created this bias corrected data file which we will utilize for doing this differential gene expression analysis. Okay. So, once we have done this, once we have loaded this, we also need the sample information which again is stored. Okay. So, the sample information has not changed, right, whether you do this bias correction or not, the sample information has not changed. So, it will remain the same, but we will have to load it and run through DSEC2. Okay. So, again going through the manual of DSEC2, I have collected the commands that we require, right, for doing our analysis and I have compiled an R script, right, which will utilize for doing all this analysis. Again this is very simple, just we look at the manual, we look format this according to their instructions and we load the appropriate data and we run the analysis. Okay. So, just going back to the bioconductor DSEC2, here is the PDF file with all the details of the manual, right, and the first thing again we need is this DSEC data set class, right. So, we need to create this DSEC data set from the data, the count data, okay, and then we can run this differential expression analysis. Again you will see there are a lot of functions that are also present in this program which we can also use for plotting our data and maybe we will use some of them. For example, the plot MA we can use for doing this analysis. Okay. So, let us use this. So, we let us load the library and also the data. Okay. So, the first step is library the DSEC2. Okay. It might take a bit of time, right, because it also needs to load other packages that are required for its run. Now, once it is loaded, we can now load the data and remember, right, we need to load the bias corrected data, right. Okay. So, again we need to change the directory to the directory where we are working. Again it will depend on your system how you set this up, right. So, here in my system I know this is where we have all the data. Okay. So, I will now load the data from here and we have the bias corrected file that we created in the last class, okay, and header equals to true. Okay. So, we also need to load the sample information file, right. So, this will be and also the header equals to true, because you have this design information, right? The bean is there. Okay, so we can simply check whether this data and sample information is loaded correctly, right? And we can then move on to the analysis now. Okay. So, the differential expression analysis, again as I mentioned, we have created a set of code which I will utilize for doing this in a very short period of time. Okay. So, we have loaded the library, we have loaded the data and the sample, we can change them data matrix and sample matrix, right, as data frame from this data and SAM, right, and then we can run these steps one after another. Okay, so first step, okay, I will convert this again to data matrix equal to as data frame. This is required because sometimes some of these tools or the commands, they expect this data in certain format. Okay. Again, this might also change from one version to another depending on the version that you are using. You might have to add a few things or write it slightly differently. Okay. We have seen this these differences between versions sometimes, and again if you see some errors, we might have to go back to the manual and see if there is some changes or not. Okay, so now we will convert this to sample matrix, right. So, frame and SAM. Okay. Now, the next step is to create this DSEC data set, right. So, we have done this before, right, and you remember, right, we use this DDS variable to actually assign this new DSEC data set, right, and the command is DSEC data set from matrix. We have count data, we have column data, right, and the designed. This is a command that we used before when doing the preliminary analysis, and we will use the same here. Okay. So, we will copy this command and I will simply run this here again. Okay. And it gives some warning message, but that is okay. We have seen this warning message, as long as it is not giving any error, it is all right. We can again write DDS, okay, and it then gives us the summary of this data set. So, this is a DSEC data set. Now, this is the dimensions are different because these are the bias corrected data sets, and remember we took the data for only those genes where there is overlap with this GC content data, right. So, GC content data and this data, our data had about overlap of about 6,700 genes. Okay. Now, once we have done this, right, what we will do, we can now run the DSEC data, DSEC analysis, right. So, the differential expression analysis and the command that we have to use is very simple, is this DSEC DDS. Okay. And we can run that now in the console, right. Okay. And okay, what I will do is I will not run this, I will first clean this up so that you can see the steps. Okay. So, once we run this DSEC command, it will go through certain steps which we have discussed. Okay. You will see this, right, one after another. It will take a bit of time, again we are running the full thing. Okay. So, here it is, you can see this, the first step is the estimating size factors. We have talked about size factors, and DSEC2 utilizes this median of ratios method for normalization. So, this estimating size factors is just estimating these normalization factors, right, and then it is calculating this estimating dispersions. Okay. So, again you remember this model, right. So, we have to calculate the gene wise dispersions, and then this is what is doing gene wise dispersion estimates. It is calculating this mean dispersion relationship, it is calculating the final dispersion estimate. So, this is the shrinkage of dispersion that we again discussed in the theory class, and finally it is fitting model and testing, right, it is doing the statistical test from there. Okay. So, we can actually get some of this idea. Again we can look at this PRDDS dollar, okay, and say sample. So, these are the samples we have utilized for this analysis. Okay. And the bean, this is the experimental design, it can give you the levels, right. So, it found all the beans and then it knows the levels are G1, G2, G3, G4. Okay. So, these are the conditions or the groups that we have created, okay, and it should look at comparison between these groups. Okay. And similarly, we can also look at size factor, and it will give us the values of each of these size factors that are calculated. Okay. So, for this sample S1, the size factor is 1.1 and so on. So, you remember the size factor, right. So, we have to divide and rather DSEC will divide these read data by these size factors, okay, which it does before fitting the model. Okay, so that's it. So, we have the results now, we can actually look at the results. Okay, so again it says, okay, class DSEC data set, the dimensions are given, the gene names, etc. Now, what we have to do is now we have to generate the results, right. So, we have kind of do this, right. So, summarize the results from here and we generate the results here. So, in this variable res, and we can simply type res and it shows you the results of this differential gene expression analysis. Okay. So, the first line you can see is the log to fold change and it specifies, okay, we are comparing bin G4 versus G1. Okay. And the wild test p value again bin G4 versus G1, the data frame has 6696 rows. So, 6696 genes and 6 columns. Okay. So, these 6 columns are here. The first column is the base mean, this is the average expression. The second one is the log to fold change, we have talked about this LFC. Then you have the standard error in LFC, then you have the statistics, then you have the p value and the adjusted p value through the Benjamin-Hochberg correction, right. So, this is the this we can then use for FDR correction. We can choose, let us say, cutoff of 10% and we can identify genes which show this significant p adjusted. Okay. So, these columns are given for all these 6700 genes approximately. Okay. So, this is the result that we have got. Now, you might ask why G4 versus G1, right. So, why not the other ones? Okay. So, by default, because if you look at the original data, okay, so I'll show you the data first and then I'll explain. So, if you look at the original data and if you remember the sample information file, this S1, S5, S9, S13, these are part of group 1. Okay. Then you have the group 2, then you have the group 3 and then you have the group 4. So, by default what DSEC2 will do, it will compare this G4 versus G1, right, the last sample versus the first sample and give the results. Okay. So, let us imagine you want this G4 versus G3. Okay. So, what you have to do, maybe just create a file with only G3, G4 or reorganize this data so that G3, G4 is there in the first and the last columns. Okay. So, by default when you have this kind of data, you will get this G4 versus G1 results. Okay. But then this is what we want in our analysis, right. We simply want this because G1 was the most interesting group if you remember from the preliminary analysis. Okay. So, this is good, we have generated this data. Now, we can interpret the data, we can look at some of the statistics and whether the normalization, for example, has been good. Okay. So, one of the first thing to check normalization is the MA plot, right. So, we have discussed this MA plot before, right, and we take these results and we generate this MA plot to see whether the data looks good or not. Okay. So, let us do that, let us generate this MA plot now from this results file, okay, or the res variable. So, I will clear this window so that it's you can see clearly and I will minimize because we will generate a plot now here. Okay. So, plot MA res. Okay, we can simply write this and then maybe later on we can specify the limits. Okay. So, what you see here is something we have seen before, right. On in the x axis we have the mean of normalized counts, right, in the data. So, this is average expression across all the samples and then you have the log fold change, this is comparison G4 versus G1. Okay. So, again we have some of these gray points and we have some of the blue points, right. So, gray points are the points for which we do not see significant results and the blue points are the genes for which we have significant difference in expression between G4 and G1, right. So, they are simply color coded based on their significance. Okay. And what you also notice is that genes that are low in expression or show low read count, right, there is a lot of variability. Okay. And again some of these genes they might show very high log fold change, but these are not significant. Again, because there is a very high variability between the technical replicates, DSEC cannot find these results significant. You will not find these results significant. Okay, and this analysis, the significance analysis, right, that actually depends on two things. One is the effect size, or how much different this expression is between these two conditions or two types of samples. That is the first thing. And how reproducible or how low the variability is between the technical replicates between these two different conditions. Okay. So, for these samples here with very low expression, the log fold change is very high, right. So, effect size is high, but the variance is also very high, right. So, that there is a lot of variance between the technical replicates, okay, and that is why these are not significant. Okay. So, is there any way to reduce this variance? Okay. And we have talked about one method, which is the shrinkage of LFC, right, or LFC shrinkage. Okay. And there is a function in DSEC2, right, that actually will do that. Okay. So, let us go to the manual and we will see whether we can find this function LFC shrink, okay, that we can use for this shrinkage. Okay. So, here is this function in DSEC2 LFC shrink that we can use. We can go to that page and we can see where we how we can use this LFC shrink. Okay. So, here it is LFC shrink log to fold change, right. So, this is the theory we have discussed, why you want to estimate this, why do you want to shrink this estimates for LFC, right, and you can do this through different methods, and here is the usage of this command LFC shrink. We have to use this data set that you have created and etc. You have contrast, you can use 3 different types of shrinkage method. Okay. So, again these are explained later on, right. You can see this. So, you can also use LFC threshold, which will come to later on. Okay. So, these 3 methods here they are explained, right. Here it is type. So, you have APGLM, this is a student's T prior shrinkage. Again, we are not going into the statistics, but these are 3 different ways you can do this shrinkage. We can use the first one and then you have the HR shrinkage, then you have the normal shrinkage. Okay. So, this is they are using different types of shrinkage estimators, right, and that is why they will give you slightly different results. Okay. So, let us use this APGLM shrinkage method, okay, and we will use this library APGLM. Okay. So, we need that library to be installed first before we can use this shrinkage. Okay. So, this library is APGLM. Okay. Let us see if it is installed in my system. If not, we can install this very easily through bioconductor. If it is a bioconductor package, I think it is a bioconductor package. So, we can simply search here and we can see whether this appears. Yeah. So, bioconductor APGLM package, right. So, again this you have the details, you have again this reference which you can read, which will describe the method, and you also have manual in PDF file that will tell you how to use this package, okay, and what are the functions that you can use for this purpose. Okay. So, what I will do now is I will check, right, whether we have this, and then we will run this LFC shrink function using this method and we will generate the final results. Okay. Okay. So, let us go to R and let us see if we have library APGLM. Okay. So, it is installed, otherwise it will give me an error, right, and then we can install it. And the first thing we need to do is we need to check these results, right, that we have generated, the results names. Okay. So, we have intercept, we have G2 versus G1, G3 versus G1, but we need the G4 versus G1, right. So, this is what we want to check, right. This is what we want to shrink using this LFC shrink function. Okay. And that is what we do here. You can see this. Okay. So, we have to do this LFC shrink on this DSEC object, right. So, after we have run this DSEC analysis, we will have to use the LFC shrink on that. Okay. So, this is what we are doing here. You can see this function LFC shrink PR DDS. This is after the DSEC analysis. we are using this coefficient because we are interested in this comparison, right, G4 versus G1 comparison, and then the type, the method that we will use is the APGLM method. Okay, instead of APGLM, you can try different methods and see whether there is much difference or not. Okay, so we'll run this command now. Okay, so and we'll store the results in this res2 variable, which you can look at later on. Okay, so let's run this in our console. Okay, and it will take a bit of time, and we can then check the results. Okay, it has run. It's saying like, okay, if you are using this for, then we can we have to cite this manuscript. Okay, so now once we have generated this res2, we can also check, right, through the MA plot, right, whether this method has worked and whether we see reduction in variability for these genes, right, the change in this log4 change for these genes that have low expression or low read count. Okay, so we'll see mostly changes, a large amount of changes here around these genes where the normalized count is quite low. Okay, so we can check this through MA plot. So plot MA result 2, right, this is the result after this LFC shrinkage. Okay, and you see now it looks much nicer, right. So this variability has decreased because we have use this LFC shrinkage function. So these log4 change values have decreased and close to 0 now, and for some of them it's 0, and this is actually more appropriate, right, because they are actually not showing very high log4 change. It's because of the variance that is present in the technical replicates. Okay, so this part is now good. Now what you can do is we can actually read the results, we can export the results, right, and one of the things I'll show you now is how we can actually save these plots. We have not talked about this so far, right. So we can maybe make it a bit nicer, right, before we actually export the data and then store it as a PDF file, for example, because we want to store these plots for later references, right. We may want to use in some presentations, in publications, etc. Okay, so before we do that, I'll probably make this plot a bit nicer, right, to look at. Okay, and then we can export this in a PDF file. Okay, so what I'll do is we'll start with this xlim, ylim is fine, right. So we can change the Cx lab 1.4 Cx axis equals to 1.2, right. So we are changing the sizes of the labels, right, because I think they are too small, right. And then what you can do also, we can use a main, right. So we want a title, so MA plot after LFC shrinkage. Okay. Okay, so you see this, right, we now have a title here, very nice one. Okay, now it looks much nicer than before, so we can now save this file, right, in a PDF file. Okay, so the command to do that is copy to PDF. Dev dot copy to PDF. Sorry. Yes, so this is the command dev dot copy to PDF, right. So it will copy this output or save this output in a PDF file. Okay, so this is the command. We use file. Let's say we MA plot G4 versus G1 after LFC shrinkage. Okay, dot PDF, and we can simply enter, right, and then we have to use this tape dot off. Okay, and we can now check this file whether this has been saved. Okay, and we can see, and you can open this PDF file and you can see that this is now saved in a PDF file. In R, you also have options for saving this in different other formats as images, right, JPEG or BMP, and then you can directly insert into presentation. Why is saved in PDF? Because then you can maintain the resolution, right, and you can also export later on to JPEG or BMP. Okay, there are other ways to also save in EPS format, etc., which are much more useful if you want to export them to different formats later on. Okay, so this is very useful, right? So once we have generated this plot, we can now export this into PDF file. Okay, so we have seen two types of like operations, right? Once we have generated some useful data or some results, we can export the results in text files, right, or write them into text files. And similarly, once we have generated some plots that we want to look at later on, or maybe use for presentations, etc., we can also save those in PDF format. I have shown you PDF format, but this can be also done for other formats. Okay, so there are a few other things we can do once we have generated these results. Okay, we can, for example, filter according to the alpha values, like the p adjusted values, right? We can also filter based on log fold change and etc. So this is something we can actually do right now. Okay, so one of the things we first we can do is we can generate a summary statistics here. Okay, so by just running summary res2. Okay, so what it's saying is giving us a summary of the results that we have seen. Okay, so we have 6652 with non-zero total read count. Okay, so we have this many genes. Adjusted p-value cutoff is 0.1. So this is the FDR cutoff. So 10% false discovery rate. So that's quite acceptable. Of course, you can change it, or you can redo the filtering. Then you have LFC greater than 0, up regulated about 1600 genes, so which is about the 24% of the whole data set. You have LFC less than 0, that's down regulated genes about 1378, so 21% of the data sets. You have some outliers, is a very small percentage. Okay, and this kind of gives us, okay, there are a lot of genes that are showing this up or down regulation. And maybe we can now look for only the top genes, right? So the genes that show significant difference in log fold change. Okay, so LFC greater than 0 does not mean much, to be honest, because LFC could be 0.2, 0.3. These are very small differences in terms of actual expression levels, right? But they are significant, right? But then if you have 1600 genes, you would probably be mostly interested in the genes that show very high log fold change, right? Those are probably more interesting to look at. Okay, so on top of this data, we can now use our own filtering criteria. Okay, and this is what we are going to do. Okay, and it's very simple actually. So we can create these cutoffs, right? So for example, we have we can generate these tables. We can use these LFC thresholds on top of the data. Okay, so here is this one. The first one is we are generating these results where we are using this alpha cutoff 0.05, right? So means we are now reducing the FDR cutoff. Okay, all right. So what we can do is we can set our own filters. Okay, and we can run these different p-adjust cutoffs. So we can change the FDR cutoffs, and we can also change the LFC threshold cutoffs. Okay, so we'll try that using the results that we have generated, but we then have to run this on the DSEC object, right? So after this differential gene expression analysis that we generate, we can then run these filters, and then we can see all these results. Okay, so first thing is we are actually running this on okay, so we'll run this PRDDS alpha 0.05. So this is looking at only genes for which p value is less than 0.05, right? We are filtering those, and then after that we can also see how many genes are there where p adjusted is less than 0.05, right? So we have 5% FDR, right? So so only about 2611 genes that actually satisfy this criteria. Okay, so this is something that is a significant reduction from the earlier value, right? We have seen about 24% genes showing up regulation, 21% genes showing down regulation. Here in total about 2600 genes, right? So that's we are making this more stringent. Okay, so similarly we can use these LFC thresholds, right, and we can see, right, whether we can generate. Okay, so we are using this LFC threshold of 1, right? So log to fold change threshold of 1 means we want genes only those genes which show at least twofold change in expression. Okay. So, this is what we are going to do, right, and then we can again have a look at this, right. Out of these genes, what fractions are showing this p adjusted less than 0.1. So, it is about 110. Okay. So, that is a significant reduction. Now, we can make it more stringent, and we see only 93 genes, they are showing LFC threshold of they are crossing this LFC threshold of 1, means they are showing at least twofold change in expression. In addition, they are showing p adjusted, right, that is less than 5%. Okay. So, that is a good thing. We have 93 genes, and these are probably the most interesting genes. Okay. We can do further analysis, right. We can look at some other things, right. For example, we can sort this list, we can find out the top genes, etc. But one thing we can simply do is we can simply export this data, right. Whatever we have generated, these results, we can simply export this, right, and then we can play around with this data set. We can identify which top genes we want, right. For example, maybe we want only the upregulated genes or downregulated genes, and that is something we can do, and we can analyze those data sets. Okay. So, again, for exporting, we simply have a look at res2. So, perhaps we can just export this, right, in some the genes list, and then we can do our other type of analysis. Okay. So, if you look at the genes list, of course, again we have to use these other things that we have used. We do not want quote, right, and we want tab separated file, right, and this should be created in the. So, the genes list, this is containing lists of all the genes, right, that are present in the data sets, and also the log fold change values, and with that data we can now play around, we can set our own thresholds, we can do different sorts of plotting, etc. In the terminal. So, one thing I will just do is finally is have a look at the file, right, that we created, this d genes list file, and this is what we get, right. This is the final outcome. All the genes are here. You can, if you can go to the last line, you will see we have 6,696 genes, right. So, total of 6,600 with the header, we have the extra line. We have the average mean expression. This is the first column here. Then of course, after the gene name, then you have the log to fold change value, you have LFCSE, you have the p value and the p adjusted, right. So, these are the two values that we want to work with, for example, the log to fold change value and the p adjusted value, right. These two are the most interesting ones for us, and we can do further analysis, plotting, etc. with this data once we have generated this file. Okay. So, this is the first part of the DSEC analysis, right. The next part we will use is that we will utilize a data set where we have the spike in present. Okay. So, we will have this spike in mix with which are synthetic RNA molecules which have been added to the samples, right, in equal amount, and based on that we will do normalization and we will run differential gene expression analysis. Okay. And so, that will be for the next class. Thank you very much.